So Jess practices listening to and reading surrounded landscapes while gardening and wilderness guiding. Jess earned herbalism, nature awareness, outdoor education, and permaculture certificates, a BA from Cornell University in ecological literature with Native American Studies minor, an MS in environmental studies with Vermont Middle and High School science teaching licenses from Antioch. And an MS with a concentration in ecological landscape design from UVM. Jess is a UVM alum. She has served as a nature mentor, wilderness guide, university farm co-manager, Waldorf Earthcrafts educator, public school science teacher, environmental studies college adjunct, university guest lecturer, scientific researcher. As you can see, a lot of acolytes. We are very proud of Jess. She does really amazing work consistently. I don't know how you have the time in the day. As Joseph said, it's been a really wonderful time getting to know you, and I'm excited to see what you've been up to next. And yeah, take it away.
So good afternoon, everyone. Happy November. A little seasonal warm. I don't know. It's the new normal. Who knows? So really grateful to be able to share with the department some of the work that we've been able to conduct, and very grateful to be part of this community. And this is Kat O'Neill.
Yes, hi, I'm Kat O'Neill. I am a recent graduate of the plant biology program here at UVM, and I was the Myco-Phytoremediation intern for this month.
Great. So I'm going to be presenting most of the information, and then Kat will come up and share some tidbits of her experience. And it was amazing. Kat was such a solid part of our team, so we are very grateful to you. So as mentioned, I work in the department as a Myco-Phytoremediation technician, and I also run a business called Myco-Evolved, which is part of the reason I came back to UVM for second master's, because I needed more training. And I want to thank Joseph Gores for mentoring me throughout this time period. And today I'll be talking about a pilot study that then led to this study. You can see a picture of this buffer that was recently restored this summer. This is Carol McGranahan, an Abenaki elder who we are grateful to have collaborated with. And she is holding a butterfly here next to a land acknowledgement that she wrote, reminding us that we are on the traditional ancestral land of the Dakina, the Abenaki people, and reminding us that our job is to be stewards and honoring the many people and lifeways that were here before we were here, before the systems that we know are here, and actually before the ecosystems that we are working with were here. And you will see this as a handrail throughout the presentation. Shelburne Farms is hosting our research, and we aim to support their mission and vision. We take their definition of sustainable a little further in our hopes to move it towards regenerative and really rooting in stewardship and community. One of the things that we've been working on with the participatory action efforts that you will see in agroecology is working with the farms and the other collaborators that support missions already began. And so the words in bold are ways we see our research can fit into what they're already doing in terms of culturally responsive practices, inclusive practices, and boosting education. One of the things about Shelburne Farms, it is kind of a classic example of what many farms in Vermont inherited. And one of those things is a colonial history of ecological and social imbalance, and more accurately, destruction. Legacy phosphorus, which we'll learn more about today, and current inundations that are still flowing into the lake as we speak. There are areas out of production that are very challenging to manage, such as this tunnel trail. Non-native species in great abundance. And then certain features that are part of the farm systems that will not be easily changed. And that provides opportunities for people like us to come in, bless you, and demonstrate solution-oriented practices. And it really needs to come from people outside of the farm, because farm staff are pretty maxed out. And good for folks doing that to have humility and patience, knowing that integrating our findings into complex farm systems takes time and might not happen during our lifetime. Maybe it will. So the crux of this research is really about watershed health. We go back to the grandfather of colonial wildlife ecology, Aldo Leopold, in the 1940s, was already pointing out that worldwide freshwater health was threatened by human terrestrial practices. Fast forward 3 to 5 decades, and we have this metric of a 50% decline, and that was 5 decades ago. And so that metric is probably much higher. The primary threats to fresh water remain consistent: land use disturbance, altered hydrologies, and non-native species presence, all of which you'll see at this site today. Phosphorus, as we are continually reminded of in this state, is threatening the health of the lake. Right now, we are only focused on the 38% coming from agriculture. And other people, thankfully, are working on the other 52%. And it's clear that basically the cyanobacteria blooms that we see when you have beach closures are directly correlated to the excess of phosphorus in the waters, facilitating the growth of algae, and that's taking away the oxygen from other aerobic organisms and also providing threats to health of any organism that enters the waterway. So I'm going to bring in the potential main characters of the research. We're still trying to understand their role. Mycorrhizae fungi are a category of organisms that have been on the planet 407 million years ago, entered the scene in the Devonian era, which was when plants were moving from water to land. And we know this from fossil records that in the roots of plants found this organelle and arbuscle, indicating that these fungi were living in these primitive plants. Fungi had been on the planet 1 to 200 million years previously and already had very robust skills at mining nutrients and minerals. So it makes sense that in this nutrient poor, thin soil, these new plants emerging from aquatic environments would benefit from a partnership with these fungi. And I find it interesting that they assisted plants from water to land, and now we are considering partnering with them to protect water from practices on land. So I'm just going to break down some jargon terms. In the scientific field, we are working within the ecological restoration field, which has been only around 50 years. And within that, the subfield is myco-phytoremediation. Working with fungi to clean up toxins is what myco-remediation is. In this particular case, we're working with mycorrhizae fungi, category of fungi I just described. And of the 7 types of mycorrhizae, we are working with 2 different types, arbuscular and ecto, otherwise known as endomycorrhizae. They're most commonly found in farm agroecosystems. The other piece of this is working with plants. So the idea is that the fungi expedite the movement of phosphorus out of the soil into the plants, and then you practice phytoremediation. So now the plants have phosphorus in their above ground bodies, and we're harvesting those plants, which is a form of phytoremediation called phytoextraction. At the time, the maximum phosphorus is in their above ground body. Put the 2 together and you have this term myco-phytoremediation. And this relationship between the fungi and plants is shared among over 80%, play a key role in terrestrial ecosystems, regulating nutrient cycles, influencing soil structure, and contributing to ecosystem multifunctionality. If you're not familiar with the relationship, plants practicing photosynthesis are basically bringing that carbon down to their root zone, the rhizosphere, and feeding the roots, the fungi that are in and around that, extending it 10 to 100-fold, mining formerly inaccessible nutrients and water and giving that back to the plant. It's not always mutually symbiotic. It can be parasitic. So this man, Michael Phillips, at this point is now an ancestor in the regenerative agriculture community. This was one of his last offerings to us, was this book. And in it, you will see a well-known fact about the ability of mycorrhizae to expedite phosphorus acquisition 4 to 100 times. Just to take a quick peek, this is what an arbuscle looks like. This is what a similar organelle, this organelle was found in the Rhine shirt. These are vesicles that we use when we're doing root counts. So when we learned about this, Dr. Joseph Goris and I published this this literature review, basically summing up all the mesocosm and field studies we could find that were working with mycorrhizae. And basically the gist of all the studies were saying there's high potential that this can mitigate phosphorus runoff into water, and that working with the plants is a very important piece, that the phytoextraction piece is critical. And so that gave us enough kind of, I say, research gap identification to then begin mesocosm studies and field studies. So I'm now going to jump into site history of the area we'll be talking about. This is pre-colonially. After the glaciation, the Abenaki were moving seasonally. Pollen core studies indicate the succession, from rock to lichen, primitive plants all the way to mixed hardwoods had robust endo and ectomycorrhizal networks. And the forms of land tending the Abenaki practice, what we call polyculture, cropping today, and permaculture, were well underway, and these nutrient exchange networks were above and below. Fast forward to now, we have countries and states and watersheds, sub-watersheds. This is where we are focused on today, Lake Champlain watershed. This is a picture of Orchard Cove, the water body, the particular water body we were focused on today, in the 1840s. And I'm briefly just going to go through the colonial history. And one thing to mention is, if you imagine how all of these practices contributed phosphorus to the soil, and basically contributed to what we call legacy phosphorus. So early 17th century, 90% of the Abenaki are killed in settler wars and diseases. And then their land practices were replaced by colonial land practices of cutting trees. They thereby cut the hosts, so the networks died. Then you have the sheep craze in the 1840s, and sheep release a lot of phosphorus into the soil through the feces. With the refrigeration of dairy products, then you have double and tripling herd size, more phosphorus going into the soil. Then you have more fragmentation of the soil through roads, ditches, and putting in tile drains. Revegetating was not best practices at the time. And at the time, Shelburne Farms called themselves a model farm. They don't anymore, but they definitely were open to subsidies from the USDA coming out of World War II technology to apply superphosphate fertilizer. And then Dutch elm disease took the remaining big trees that were on the landscape out, and then non-native species like buckthorn, honeysuckle came in. So this is the area we're working on right here. Due to the geologic history, it's glacial-lacustrine silty clay soils, i.e. Covington clay, highly erodible. This was in pasture for decades, tile drain networks underneath here. And when we took water quality samples, the water was 18 times what Vermont water quality standards say any water should be, leaving a tributary entering the lake. So current site challenges, just to name that there's a lot of buckthorn far beyond the area we cleared, and the vectors of rodents and wind and birds is humbling reality. The compost pile, 550-yard cubic pile, is currently there, and to our knowledge, not going to be necessarily moved anytime soon, despite a report that we submitted approved by UVM Extension to create a vegetative treatment system below with suggestions to move it. But again, remember that site condition I mentioned at the beginning with very hard to change farm systems. So that's a place where I just say, hey, FYI, this project's a little bit like a Greek myth. Is it Sisyphus pushing the rock up the hill? If you're not turning the faucet off, we're never going to remediate the site. So this is actually a demonstration project. Even if you turned off the faucet and you removed the pile yesterday, research indicates it would take 6 to 9 decades before you would actually see statistically significant decreases in phosphorus, because phosphorus is very slow moving. And then a lot of the tile drains are broken, they're unmapped, and we found a few in different parts of the sites, which would create some soil variability in our testing. So I'm going to briefly run through the pilot. This was basically our first attempt to try to create a interception between, I guess I can go back and just say, this is the interception area. This is what's called the critical source area, the pollution source, and the lake. So we tried to put it right in between. And you will see throughout the presentation today, RV represents uninoculated restored. It'll be in red. RVM is restored vegetative inoculated with the mycorrhizae, and OIV is the control. And so we removed buckthorn from 2 areas. We inoculated one of them. We left the control. And the objectives of the study were to reduce legacy phosphorus for the reasons already shared, increasing pollinator habitat because we're currently in the 6th grade extinction, and entomologists like Doug Talame suggest that if you can introduce hosts of pollinators in any area that's not in actual production, you can reverse or at least slow down the current extinction. And then support rematriation, because to us, at this point in time, it does not seem that we can just focus on an ecological solution without the social imbalances that accompany it. And providing access to the original people's plants and lifeways is actually part of the solution. And this is a view of the buffer this summer. It's really beautiful. We actually took the fences down. So if you ever want to see what a 6-year restored buffer looks like, check it out next spring. So I'm just going to briefly run through the literature that we published from the study. The first is this paper in Restoration Ecology, which just has 2 years of data in it. And basically, we saw in that case that mycorrhizae appeared to reduce soluble reactive phosphorus concentrations, and 1.7 times more species appeared than were planted, which indicates something called autogenic succession, which is this idea that, you know, we try to help and then we get out of the way, and the systems can take care of it. Recommendations from this are something that most scientists are not yet doing, of gathering pre-colonial site history and local original people's knowledge to inform the design process, apply observations of local site-specific riparian buffer polycultures through walking and paddling different waterways, and then thinking about original people's access and use in mind when you're choosing your plants, and then inoculating the plantings with native mycorrhizae from your nearest undegraded areas, applying manual labor rather than chemicals. We learned a technique from Mike Ball of Got Weeds. 3 times cut in 2 seasons creates a 90% death rate. No need for glyphosate. And then cyclically coppicing those woody stems and harvesting the berries in late summer, which we found is really the second week of August, before senescence, right? So before, a couple of weeks ago, when the leaves started to turn, because when that starts happening, all of that phosphorus that's uptaken in the plants is going to go right back into the soil and water. You were able to obtain funding to then monitor this for 2 additional years. And this early winter, we published this paper, which is now 4 years of data, with Carol McGranahan. And the findings from that were that 58 newcomers in terms of genus species showed up. So you can really see the succession further along. Total phosphorus decreased over in all treatments. The greatest, however, was in the uninoculated plot, which is a head scratcher. And if we have time, we'll get into it. But that informed the new study we're doing. We have 3 hypotheses about why that is. And that while biomass and P biomass of the plants did not differ in treatment, they did among species, which is really good data for going forward of applying these. Elderberries have the highest concentration of phosphorus, over 3,000 milligrams per kilogram. And then you can do back of the envelope calculations. 0.1 to 0.5 kilograms per hectare per year might be low because we were only harvesting a couple of the species. In terms of, if you imagine a drone over the entire lake, all the point and non-point sources where phosphorus is doing this and harvesting, you could actually have a fair amount of potential phosphorus mitigation, preventing it from going in the lake through the harvest. So our recommendations, this will be redundant from what you heard. I'll try to just highlight the new parts of it, that Abenaki harvest can remove phosphorus and be an effective form of phytoremediation. And this is work the farmer does not have time to do. One of the things is the Abenaki practice the honorable harvest. You may have read about that in breeding sweetgrass, and that's only 1/4 of the population. So there's a balance between protecting biodiversity and phosphorus mitigation. And we're not removing as much phosphorus, but we're protecting the biodiversity. So that's something to balance. And then co-designing using the endemic mycorrhizae and applying these cyclical harvests. These are strategies we feel that can not only help with water quality, but also pollinator habitat and the pollinators themselves. So I just want to take a moment to honor Rhamnus cathartica, buckthorn. It's never, I feel, good practice to malify any plant. I don't like the term invasive. And I've seen what they do in ecosystems. I know that many of them have been identified as ecosystem placeholders, as well as providing medicine for today's modern diseases. And I'm going to point out reasons why we feel justified, with humility and knowing, you know, in many years we may stand corrected, of removing it in this case. And why it's present indicates degraded habitat. So if you have not witnessed them before, you will see that the early growth shades out ephemerals. The Latin name cathartica refers to the laxative effect they have on the birds and the berries. The beautiful flowers only provide pollinator habitat for 2 weeks, whereas a lot of native species provide habitat for pollinators for much longer. The mycorrhizal networks really limit the diversity of plant species that are growing underneath. And in our earlier studies, we analyzed phosphorus uptake in the plants. And you can see compared to elderberry, meadow willow, red osier dogwood, and arrowwood, buckthorn uptook the least. And it's not providing medicine or food. However, you could use it for firewood, mulch. People at Interview are experimenting with saprophytic fungi growing on it. And many of the patrons at Shelburne Farms love the magical tunnel-like effect. So as a restoration ecologist, I'm also responsible to the riparian practitioner community and kind of finding gaps in their practices so that this research is relevant to them. And what we found is that basically right now, the way that riparian buffers are planted and kind of funded throughout the state is not really with opportunities for gathering data or maintenance. And so that means that their function of uptaking phosphorus and removing it is moot, because when senescence happens, it's all going back to the waterway. People who are designing the palettes and choosing species are not thinking about pollinator habitat in their choices, nor Abenaki use and access. And when I'm in a room full of practitioners, I'm usually the only one that's not using glyphosate. So we feel that our strategies have a solution in those gaps. And some people will say, well, what about 6 acres? You're going to do it all by hand? And we have an answer to that. We did use some machinery, and we have metrics on that. So we feel that it can be applied in a larger scale. No need for glyphosate. So you're going to recognize that the first 3 objectives of our new study are same as the pilot. And the additional 4th one is fostering eco-literacy. So when that study was completed, colleagues suggested that we improve the study based on our findings, and instead of doing it through UVM, do it through the business I run, Myco-Evolve. And in that, UVM has been a very solid partner and an incredible support. We're very grateful for that. One thing I just want to share, this picture I love, was early spring after planting. You can see this polyculture of Joe pieweed. This is baby elderberries, willow, and then the red clover. And so we're just excited about how the palette is looking on the landscape. So a map is the original study I mentioned, and above it is where we put in the new study this spring, basically tripling the plots, random block design. Instead of commercial mycorrhizae, we used endemic mycorrhizae. We have much more robust data sets, and instead of the original plant palette being 88% edible, medicinal, utilitarian to the Abenaki, it's now 98%. This is just a picture of the concept map, and everything is planted based on where the waterway is. This is the field and where the phosphorus is coming in. So the hyperaccumulators of phosphorus are closest to this area, and then based on microclimates and companion planting. This is a bigger view of the actual palette, and you can see in it, along with the common scientific names, the Abenaki uses, edible, medicinal, utilitarian, or ceremonial. You see the types of mycorrhizae, AMF or ECM, some like willow or both. This is the window of pollination from March through November. And then these are the hosts that the plants attract and provide habitat for. Just a brief note on mycorrhizae that's endemic. So through another study with a wonderful intern who became a research assistant, Luca Kolba, who was a student in Rubenstein a few years ago, we were able, through a SARE grant, to do research at Diggers Mirth at the Intervale and publish this free SARE, a guide on the SARE website of how to grow endemic mycorrhizae. There's also a YouTube video about it on our Myco-Evolve YouTube site. And it can be done in 16 weeks. It's pretty easy. It's pretty low budget. You will want a lab for checking viability. And basically, the farmer took us to the most wild part of Shelburne Farms. We filled 2 5-gallon buckets of the soil. And then in the soil lab in the greenhouse, we grew it out. We tested the roots and looked at the spores for counts to check viability. So that's what we used in the new study. And we gifted extra to the farmer. And this is kind of application. I'm just going to briefly run through the methodology of our preparation before we look at data. So we pasteurized 7 yards of low phosphorus medium that was a mix from Vermont compost. And we potted up at the UVM horticulture farm 2,200 bare root plants. Half of them were inoculated with mycorrhizae, the ones with flags. Meanwhile, a chainsaw crew from VYCC came. Here we are in the spring planning. This is Dana the farmer, VYCC crew leaders, and Vermont Youth Field leader. And we had 20 work parties removing buckthorn by hand. As you can see, it's a lot of work. And that is how everything was prepared. And then we had to loosen up the soil with broad forks. We only removed the buckthorn away from the waterway. Near the waterway, we just practiced the strip back 3 times in 2 seasons approach so as not to disturb the soil and release phosphorus into the waterway. Covington clay is difficult soil to work with, so it took a while to loosen it up, leaving any native species like these red cedar and the dandelion. Meanwhile, Vermont Youth Build put up this lovely fence. And thanks to many helpers, we laid down this choir, which we put around the stumps. And we watered, thanks to the water truck, and brought all the plants from the Hort farm and put them in their appropriate places and planted them. And here's Kat watering the nursery. Definitely watered the plots a lot. Tool maintenance was a big part of our reality. And then stripping back the regrowth. This is what it looks like, the second growth. So you strip that back, you do it a 3rd time. Most of the stumps are dead. The remaining 10% just strip back next spring. It still provides habitat for fungi, insects, and microbes, and overwintering the birds. So data. You can see that cats in all these pictures. Basically, we're using an Oakfield sampler and processing the soil for total phosphorus, malic, and soluble reactive phosphorus. Here's Kat harvesting the sweetgrass, which we dried and then ground thanks to the crop and soil lab and analyzed for phosphorus. Mycorrhizae, we harvested the roots of certain plants, dyed them, checked the microscopy. Here's the slides. And then for biodiversity, we did transects documenting the species that showed up. So now Kat's going to tell us a little bit about her experience.
Yes. All right. So I'm going to tell you guys a little bit about my experience and the different skills that I gained during this internship. And so I put 2 different sections because some of them I was expecting going into it, and others were a bit of a surprise. So on the expected side, I engaged in a lot of data gathering, and that was a lot of soil sampling and harvesting mycorrhizal roots, mounting them on slides, doing the counts for those, as well as maintaining the plots, which involved a lot of weeding and watering and just going there every day and making sure everything was in order. And I also had the opportunity to use my GIS skills to help create an iNaturalist project that people can publicly engage with to track the biodiversity that we have going on this project. And then kind of on the unexpected side of things, I was really able to engage in a lot of teaching moments and leadership moments with being able to help mentor some of the crews that came in, the volunteer crews and the work crews, as well as really honing my scientific communication skills, both through making the posters that we had hung up on the plots and just chatting with people who were walking by the plots as we were working on them. And this project also really gave me a good idea of the realities of conservation work and working within deadlines and budgets and things like that.
Great. Yeah, Kat was definitely a rock star, and we're sad but happy for you that you're going to move on to other things. And so if you know anybody who wants to do this next summer, you won't be involved in installation, but you'll definitely be involved in a lot of the field and lab work we are going to be hiring. So spread the word. And these are the plants that were harvested for Abenaki use this year. This is Nicotiana rustica. This is tobacco. Phyllis gricia ground cherry, and Hirocula odorata sweetgrass. And elderberry, this is actually from the pilot because we planted the elderberry in June, so it didn't flower this year. But we did have a Abenaki education program. This is Carol McGranahan teaching us about the plant harvest and guiding us in elderberry syrup making. So Kat is going to share a little.
Yes. So something that really resonated with me about this project when I learned about it, and also as I continued to engage with it, is how Jess is able to really practice rigorous science while also maintaining a personal, spiritual relationship with the people, plants, fungi, and the land that we are working with. And kind of an example of when this came up in our project, if you see this picture up here, that is when we were doing the root staining of the plants so we could check for mycorrhizal colonization. And that is of Nicotiana rustica, the tobacco that we were using in the plots that is sacred to the Abenaki. And what we found with that is that the entire root was stained pink when really, even though we kept our diligent protocol that worked for all the other species, we could not gather data from this plant. And rather than really pushing it to try to extract data and change the protocol for that plant, we decided to honor the special connection that the Abenaki have with this plant and kind of step back and not include it in our data set, which we found out later didn't really end up affecting the data set. So it worked out after all. But this continues to be a living edge for me, kind of exploring and navigating the boundary of where I apply science versus honoring spiritual connections with lands and planets and people.
Thanks. So on the deliverable end, it's a 3-year grant, and we're expected, obviously, to produce data to inform the required agricultural practices and to publish a manual for the state that amends best management practices for forest and riparian buffers. So we currently know that the manual will include data to inform recommendations and the multifunctional plant palette, managing through harvest, collaborating with original peoples, and non-native, non-chemical removal of non-native species. We know that that's going to be in the manual, and that will be new information that's an update to current best management practices. What we don't know, and will be dependent on our data, is really if endemic mycorrhizae will improve the actual phosphorus uptake in those plants, and thereby be assisting in phosphorus mitigation. We know that restoration itself is assisting in phosphorus mitigation in the practices, but whether or not the mycorrhizae, even though the literature indicates it will, have to see with the data. So I just want to briefly touch on our 4th objective of eco-literacy. These are ways that we have been demonstrating that. There's a curriculum that's writing currently for elementary, middle, high school, college, and graduate school students. We're providing tours and presentations, lots of work parties, and there'll be maintenance parties next year. And then Kat and I created an INAT project. These are the categories of people that we are translating our findings to. And obviously they're very different languages because they're very different groups of people. And I just want to highlight the different groups that were involved in the site and note that this is definitely a community project. There is no way that this would happen and would have happened without the 100+ volunteers that helped make it happen. So I'm going to jump into the data. And I know, for a seminar in a scientific department in a university, it's a little late in the presentation to get into the data, but this is year 1. And so, it's basically just baseline data. Mycorrhizal colonization, this is just checking to see, did our inoculation work? And we did have statistical difference, which is great. This is most visible in this figure through the herbaceous species, which you will see later in the phosphorus uptake data is really interesting. And not only statistically significant in treatment, but also category, particularly the herbaceous species, which is really new for us because in our pilot, we did not harvest or work with the herbaceous species, but literature said you should check it out. In terms of the soil data, both malic and total phosphorus, really nothing statistically significant. We didn't expect it, but you do see in both of these a trend going down. I do want to note that the statistician really was like, you shouldn't even put 418 in there because technically we didn't do treatments. But that's the plots themselves. So 418 is like pre-restoration, 6.3 is middle of buckthorn removal, and then 8.6 is after the installation. But you can see in both metrics a sort of decrease, and that's just typical of what happens with phosphorus throughout the season when plants are uptaking it. If we took samples like today, you would see the numbers going back up because of the release that we talked about. And then this is just interesting, looking at just the during restoration and post restoration soluble reactive phosphorus, there was statistical significant difference, which you would hope between the control and the 2 restored plots, not between the inoculated and uninoculated, too early to tell, but that's really interesting. It's like, okay, at least we're seeing something going on between all that work. So still not much you can say from that. In terms of phosphorus uptake in the plants, there was no statistical difference in treatment, but there was in category, which is, again, interesting. The herbaceous were much higher. And in particular, tobacco and sweetgrass. Well, those were the ones that we harvested. And again, that's new data, and that might improve our back of the envelope calculations, adding to the elderberry a lot more herbaceous species. And these are medicinal plants for the Abenaki that they're certainly going to harvest every year. So that's hopeful for phosphorus mitigation. And in terms of biodiversity, this is what you would expect. Both restored plots obviously are much higher plant counts than the controls. No difference between the inoculated. Too early to tell, but worth noting that migrated is loosely used here. It's referring to species we left in place and that came in, that half as many migrated in or were left in place than were planted. So that's showing that autogenic succession is already underway. I just want to show the faces of some of the 100 people that helped. These are the 2 VYCC crews, and these are some of the topics we talked about in the field when working together. This is Youth Build. If you're not familiar with it, it's an amazing organization in the community that funnels high school students that can't deal with traditional schooling and really activate their more kinesthetic, dynamic learning styles. And they did an incredible job helping remove buckthorn and put in these really beautiful fences. And then Permatours, if you're not familiar, is a bioregional earth repair group that came and removed an entire plot in one day. And then Myco-Lab, the community branch of Myco-Evolve. And then these are kind of offerings that we've been, sharing and continue to. Check out the INAT project, still very much in the works. And their research plots are available. Our pilot is complete. And the fence is down. You don't have to worry about hygiene footwear, which you do have to worry about in our current study. You have to change shoes going between inoculated and uninoculated, or be barefoot in one of them. But we are offering the pilot with Shelburne Farms approval because there's still a lot of great research that high school student groups or college student groups could do. If you know any pollinator researchers who need an area that's already been prepared for them, we would love to get metrics on the actual pollinators visiting the habitat. And we're trying to track down some mycorrhizal DNA because to track the actual genus species of the fungal communities from pre, post, and then a few years later is going to be valuable to the mycological community. And yeah, future research, just to note that Joseph and I are going to be, contributing our efforts to this for 2 more years, but after the grant term, we're looking to hand it off and the research questions will change. But if anyone knows people who might want to take this on and take it to a different place, that is welcome. And of course, just have to put a plug in for funding because as all scientists know, we're always writing grants. I'd like to keep my post at UVM at least during the study. The manual, we have some funding for it, but more would create more opportunity for detailed graphics. Again, if the pollinator specialist is funded and the DNA mycorrhizal specialist is funded, that would add to our data. And I just want to acknowledge the many people at UVM who've been incredible support, the Abenaki community, the regenerative community, project advisory committee, and all of the funders, particularly, just noting our fiscal sponsor, Permaculture Association Network, UVM Extension, and the ALE, the College of Ag and Life, Lake Champlain Basin, and their EPA branch. And then Grassroots funded the original rematriation ceremony we did with Alno Byway years ago. And the Lintillac Foundation has been an incredible contributor throughout the entire pilot to now. And Permatours as a partner with Vermont Youth Build and Resource. And then Vermont Ag, Food and Markets has been supporting us through Alex Sapillis, who's on our advisory committee. So with that, if you want to track the research, you can follow us on Instagram, Facebook, or LinkedIn. And we would love to hear any questions. Thank you. Yes.
So I might have missed it. So you looked at the phosphorus concentrations by plant species. What those were the harvested portions of the plants, or like?
Yeah,
Oh,
yeah. So the above ground part of the plants is what we
Okay,
harvested
the whole
and
above
then
ground, or-
dried them and ground them and then submitted to the lab for TP.
Okay,
Mhm.
great.
It kind of varied, like which plant, how much we harvested. For example, sweetgrass, we did harvest the entire above ground part versus like willow, we just kind of coppice the top portion.
Yes.
And you may want to also add to this that the reason, I mean, you could dig out the whole plant, that would be disturbance, but it's also not what the Abenaki would do for harvest, correct?
It's a good point. A lot of the plants that are listed as edible and medicinal and utilitarian, we didn't harvest and analyze. And some of them, the roots are valuable, like gravel root, and like swamp white oak. I don't know if the acorns are going to be available during our time. And those are probably a big phosphorus. So I do think that's great areas for future study, that if you had a huge amount of root plants, harvesting some, that probably has a lot of phosphorus in it, but that'll be for the next people. Yes.
A quick question. I'm intrigued by the fact that Shelburne Farms was agreed on with this because they're aware that probably their practices are influencing the phosphorus. So I wonder whether now that they see that there is some improvement, I mean, it's still a short period, right, before you can really conclude, but would you think they would that it can change their practices in some ways, and also their relationship with the Abenaki people? And
Yeah, it's a great question. And by the way, Gabrielle contributed a ton of time during the pilot years, during COVID to show up. So yeah, I guess I'll say a couple things about that. Shelburne Farms is very committed to supporting research, and they're very committed to doing what they can to improve their systems, and they have very complex operations. So they reminded me to not say they will not move the pile. They might in the future. They reminded me to say that buckthorn is something that will be part of their landscape, and they're not going to eradicate all of it. And they have this program, which is an action plan to get to net zero. And so I'm presenting to them in 2 weeks, and my hope is to share this idea. What about an action plan to help Shelburne Farms reduce phosphorus? And that we could get the data on what we're doing, and then they could try to link up with the Vermont Pay for Phosphorus program. And that way they could actually obtain a tiny income source through this phytoremediation part, and maybe even moving the pile. So that's kind of a strategy that we're considering. And there's another, I think there was another area that we were thinking of. But yeah, I guess I'll just stop there. Now, in terms of the Abenaki, it's pretty complicated. They are currently working with Abenaki, and they're working with a different group of Abenaki than we are. And so that's something we are going to have to figure out because of the complexity of Abenaki politics. So what we're going to do is we're taking that objective of Abenaki, access, and we're kind of presenting it in a lighter form of how it could be shared along the whole watershed. So in New York State, with the Mohawk people, you could design a palette. In Canada with the Odanak, and kind of, we'll have to figure out how much will happen when we're gone at the plots. It's not certain. But we hope that it's going to contribute and link into their educational programs and outlive our time and be a feature of the farm that carries on their mission, and whatever researchers and people are there, that it continues to support their mission. That's the hope. Yeah. Yes.
And maybe it can become a model of how to make that connection between the indigenous people and the researchers and the farms. That would be really cool too. I mean, you mentioned the Mohawks, and Abenaki. So,
Yeah, that's the hope. And it's a trend happening right now, right, in the fields. Yes.
Have you socialized this with other farmers, and if so, how has that been going, and any response from other farmers in the area?
Yeah, it's tricky, to be honest. A lot of the production farms, like at the Intervale, besides being slammed by losing their entire production flooding, aren't able to really do this, just due to their practices. So we haven't yet found any farms that have, buffers that aren't already like, yeah, we haven't ever found farms that have a similar enough situation to share this with. We have shared it with farmers who are interested in some of the strategies. But that is kind of our work of the next 2 years, is to figure out, like, how can we offer this to farmers, maybe through NOFA. Any other questions, insights? Yes.
I'm curious to build off of that, what, and I understand this is probably getting more complex than we have time to talk about right now. But what about other landscapes makes it difficult to apply this? Is it specifically their actual like geographic landscape? Is it their relationship with the community? Like what kind of, I'm curious about what kind of headway or what kind of things you're butting into.
Yeah, I mean, I'd say right now, most of the farmers I know are so busy with just trying to survive economically. And Shelburne Farms is unique because they have this extra objective of being a research farm and a sort of, I would say, cushion in the sense that we're not disrupting their systems by doing what we're doing. And I haven't yet seen a farm that has as much land and space where we could do that.
Okay.
But I'm sure it's out there.
Okay, yeah.
Yeah.
Thank you.
Is there anyone online that's asking?
I haven't seen, I've been trying to keep my eye on it and I haven't seen any at the minute.
Any questions out there? Well, it doesn't look like there are.
Laura.
Oh, it's Laura's typing. Thanks, Laura. Great. All right. Well, thank you so much, everyone, for coming, and thank you for all the people who have supported this work. Feel free to share it with your classmates and professors, and reach out to us if you have questions. We really see this as a community project, and like we want it to get into the hands of the community and it to go where it needs to be helpful. So feel free to be in touch about that. Yeah. Thank you so much.
Can we, again?